Hi everyone, this video is a tutorial for SOAR, which is our spatial transcriptomics database with disease mechanism exploration and drug discovery capabilities. This tutorial will demonstrate the comprehensive analysis functions offered by SOAR. When users click on the Data Browser tab, they can see the 2,800 samples contained within SOAR and filter for samples here based on their research interests. The metadata and source of each sample is listed in each row. Let's first take a look at this first breast cancer sample by clicking on the sample number. Upon clicking on the sample number, a user can scroll down and view the spatial variability of expression in genes of interest. For example, let's explore SPP1, a marker for tumor-associated macrophages and which helps malignant cells avoid immune surveillance. We can see that this gene shows higher spatial variability at the bottom left and right corners, as evident from the red colors. The opacity of the spots can be lowered if users are interested in viewing the morphology of the spots. The table below can verify the spatial variability of gene expression across different cell types. Multiple gene inputs are allowed for easier comparison. Let's compare the spatial variability of SPP1 and another gene, CXCL16, whose role is the opposite of SPP1, as it can recruit immune cells to sites of damage. Once sorted by Q-value, as I do here, we can see that while CXCL16 is uniformly expressed in dendritic cells and monocytes, it shows significantly variable expression in other cell types depending on the cell's spatial location. We can further leverage the cellular deconvolution tab, where each spot is represented as a pie chart of deconvoluted cell type percentages to better understand the spatial variability. For example, clicking on the download button allows for a larger visualization here. We can see that the bottom left and right corners that have high SPP1 expression indeed overlap, with higher composition of malignant cells and macrophages. We can also see that the region above the bottom left corner are spots that contain more CD4 T cells. Let's explore another brain dataset to visualize the spatial clustering function. With this coronal forebrain sample, we can see that there are 19 distinct spatial domains, which represent individual layers in the brain. And users can further explore the difference in gene expression across these clusters. The next tab, Gene and Cell Analysis, allows users to investigate cell type-specific expression pattern and cell-cell interactions of specific genes. Upon entering a tissue and species, users can then select a gene, as I do for SPP1 here. The first Spatial Variability tab allows users to conduct meta-analysis of spatially variable genes across different samples. Red tiles represent significant results, whereas white represent non-significant ones, and gray tiles mean gene expression is not measured in the cell type. For example, we can see that SPP1 here tends to have significantly spatially variable expression. If I scroll to the bottom to these two columns, and these represent endothelial cells and macrophages, as well as fibroblasts and malignant cells. But we don't observe the spatial variable expression in B cells, CD4, or CD8 T cells, or other myeloid cells. This result concurs with SPP1's role in promoting cell proliferation and migration in the tumor microenvironment. The next tab, Neighborhood-Based Cell-Cell Interaction, shows the gene expression in the cell type of interest. In this case, macrophages, located near or not near to other cell types. Significant results are shown as larger tiles. Red color represents positive log2 fold changes, whereas blue is negative log2 fold change. For example, we can see that SPP1 in macrophage has higher expression when they are near malignant cells. Let's take a look at the last tab, which is Distance-Based Cell-Cell Interaction. This models the relationship between the expression of a gene in a cell type and its distance to other cell types. Significant results are shown as larger tiles like before. Red color represents positive derivative for the relationship between distance to a cell type and gene expression. For example, we could see that SPP1 expression in macrophages shows the greatest positive relationship. If I scroll to the bottom, with their distance to endothelial cells and malignant cells, as well as fibroblasts here. This result concurs with tumor-associated macrophages' role in promoting uncontrolled growth in these cell types specifically. Let's take a look at the last tab, which is the drug discovery function. Drug discovery function allows users to identify compounds with potentially therapeutic effects for pathological samples contained within SOAR. The perturbation effects of these compounds on cells and genes have been experimentally tested by CMAP by the Broad Institute. The master table allows for filtering by tissue, condition, compound of interest. Enrichment score represents the overall perturbation effect a CMAP instance has on the differentially expressed genes of a cell type, in this case filtered to be malignant cells. Positive scores mean repression of differentially expressed genes, and vice versa. The CMAP instances can be mapped back to the published CMAP dataset for in-depth analysis on experimental conditions. Non-filtered results can be downloaded at the bottom. Let's sort on enrichment scores and click on the breast cancer sample for the row of everolimus, which is a repressor of the mTOR complexes. Before delving into the specific compound, we can first investigate the differentially expressed genes in malignant cells or other cell types of interest, which helps us to identify gene targets. For example, we can see that MHC, HLA genes are downregulated, helping with escape from immune surveillance. ERBB3, or also known as HER3, an upstream gene of mTOR, is upregulated. On the right, we can see the protein-protein interaction in the cell type of interest, in this case malignant cells. Color of the nodes represents the degree of differential gene expression in the cell type compared to others. We can see that ERBB3 here interacts with HLA genes. We can also see that CCND1 here interacts with its suppressor CDKN1A. High expression of CCND1 and low expression of its suppressor are markers of uncontrolled cell growth and differentiation. On the bottom left panel, we can investigate the top and bottom 200 compounds in terms of enrichment scores on the cell type of interest, in this case filtered to be malignant cells. Alternatively, if we are interested in compounds that can promote the expression of differentially expressed genes in immune cells such as CD4 cells or B cells, we can then look at the list of positively enriched drugs instead, because these can enhance the differentially expressed genes of these immune cells. The bottom right panel allows us to investigate the gene targets of individual CMAP instances, such as everolimus in this case. Node colors represent the direction of log2 fold change of the gene, and arrow color represents the direction of perturbation the compound has on the gene target. Top and bottom 30 targets are shown. We can see that everolimus strongly promotes the expression of the downregulated CDKN1A in malignant cells, which can promote cell cycle arrest and slow down progression. Everolimus can also downregulate the upregulated MYC and SLC5A6, which are responsible for oncogenic metabolism. The diagram helps to explain the strong suppression everolimus has on malignant cells, elucidating its mechanism of action. This concludes the tutorial for SOAR. For specific inquiries, please contact us by clicking on the Help button. Thank you.